I'd be curious about what all organisms are also in those. So Volcanodoc last week, there's the Volcanodoc's command. Hi everyone, do you love streamers? Do you love geologists? Do you love volcanoes? Of course you do. Please go give awesome and amazing Volcanodoc that follow. We were talking last week, Volcanodoc, about extremophiles in the Arctic. And that they were incredible because you couldn't culture them in a lab in normal cell settings. You had to culture them in these extreme ice containers and all that jazz. And it would be really amazing to figure out if on these volcanoes, like in those glaciers where you're already having the extreme heat thermophiles, if there's also some like these extreme cold like microorganisms, and are we killing them off as well, like with the heat? I mean, obviously we're doing a lot of damage with climate change, but it's just something to think about. Agar does not work, Mike. So they took like high nutrient agar based media and like the agarose dish, like the squishy that you see like E. coli grown on, as well as cell culture media. Cell culture media is a liquid. It basically has growth signals as well as a ton of nutrients. And so those, like when you culture cancer cells, those are not on an agar plate, those are in a dish with cell culture media. And so either one of those are meant to grow organisms. So like cell culture media, in fact, is notorious for like getting contaminated with outside bacteria. So in theory, if it was just nutrients that these extremophiles needed, if you put in the cold-loving bacteria, it should be sufficient for them to survive. If all they need is food, but it turns out they need like the external cues and smikes and Volcanodoc. I'd love to know your opinion on this. The extremophiles that live in the ice, it turns out you need to culture them in groups. If you isolate a bacteria or like one bacterium, they don't grow. Unlike E. coli and yeast, you put one single one, it grows and takes over the plate. These ones in the Arctic don't. And I don't know, Volcanodoc, do you know if the thermophiles are like that, where there's a lot of in-between signaling between individual cells and that signaling required for their growth? I might be going off totally on the deep end on that, but I would be really interested in it. Probably some weirdo extremophiles in volcano glaciers, says Volcanodoc. They would have adapted to tolerate toxic gases that are emitted out of the volcano. Ooh, see, that would be really cool. All right. Let's actually continue on that front of like rock and minerals. Because we have Volcanodoc here, let's continue with rock extremophiles. How about? From a newbie perspective, I mean, they're probably oversimplifying things for them to be relatable and understandable by us laymen, but they're good. Thindall. So oversimplification on its own, I think, is a hard term. There's a delicate balance, right, between it being patronizing and just like you are just being a jerk versus helpful. And so what level of simplification is helpful but still conveying the correct science behind things? And then, because if you establish a groundwork of simplicity, that it's like you're not talking down to people, which is, I think, a big issue in science, right? They're like, oh, this is a layperson, let me dumb it down, quote unquote. It's like, that's not what you should do. You should just be a better explainer, number one. And then number two, like, some of the concepts, you can skirt around and just be like, okay, like CRISPR, you cut out a genetic element and you put something in that you want. That is, 10 years worth of research right there that I summed up in a sentence without any detail, right? As long as you can follow it along, and then you might be like, okay, well, how does that happen? And then we can go into it deeper, right? Is in my opinion. Simplify something without lying, but also not giving the details that require 5 years. Yes, exactly. But no, the reason I mention is because a lot of scientists will do the patronizing oversimplification, which really grinds my gears. So that's why I'm always like, keep the balance and make sure you're being helpful and not a jerk, if that makes sense. And the interesting point is, again, a lot of these, if you tried to culture in a lab under non-tough conditions, they don't thrive and they don't do well. They actually get there was one study that I was looking at because I thought it was a really, really cool concept last week about the Antarctic ones, where they called it nutrient shock, where the organisms were placed in like regular nutrient media for normal cells, but it was so high nutrient for them that it ended up killing them. And what's cool about that is that actually is a model for aging. So it turns out that they've done mammalian studies on this too, but a lot of it in insects, fruit flies, C. elegans. If you starve or calorie restrict animals, they're more likely to live longer. And so these extremophiles are overloaded in nutrients when you're putting them to regular dish and killed instantly, which is kind of gnarly cool. Morbid again, but is there any way related to what happened to CC prisoners after World War II when they couldn't be given unlimited nutrients because it shocked their system and they die? It probably is Thindol. I mean, Thindol, that's the same way that addiction behavior works, right? So if you're trying to wean someone off a medication or a drug, it doesn't have to be a drug. It could be medication as well, right? So like antidepressants, for example. If you are instantly take them off, there's a huge negative detriment to your health. That's why you have to wean off them very readily with your doctor's help. Same thing with food. If you are been starved for so long, you will undergo a food shock if you're brought back. So actually, Thindol, my grandpa was at a POW camp, and when he got out, he was, I think, like 90 pounds from the POW camp, and he's one that also like had to be, to use Smike's terminology, titrated back on for food because of how like thin that he was. So it's probably like it has to do probably with your metabolism, and also I think with humans too, you can go into like an insulin shock because you've been so deprived of sugars as well that you're just, how your insulin's working isn't primed for having anything like that, which is crazy cool. Java knows where we're going on all these things. Hi, True! Happy Saturday! Java's like ousting it, because guys, where we're going to take this eventually is aliens. But yeah, we're talking about extremophiles, and in particular, we were talking about them from the perspective of just living in like different locations on the Earth that are extreme, and that allows them to maybe be the reason why we could have life outside the planet Earth as well. Because if we can find living things that are on our planet in these insanely unhospitable environments, the chances are that maybe there's some kind of microorganism living on a different planet as well. Alrighty, let us continue on. We're going to go deep sea. We're going to go deep sea to start with. And then we will go to possibly the tardigrade. And we might do or I don't know, do you guys want to talk about the space? And we're kind of on the topic of the panspermia. Maybe let's just direct to that, and we can save these other animals and extremophile examples for next time if we so choose. On top of that, too, remember we just talked about an extremophile that survives in the ice in the Arctic, like pretty readily. So it doesn't have to be necessarily just in the water.
The spell is broken. We live again.
Hi, KT. Hello. What is making your head hurt? What is the cutoff point for being called an extremophile? I mean, from my point of view, I'd say pretty much we consider penguins extremophiles, or pretty much everything in the deep sea. So that is a great question. I used to think it was only like single-celled organisms at places of extreme salt, temperature, or like pH and phosphorus and those other mineral concentrations. It turns out pressure is also a big component, and so deep sea organisms do count as extremophiles. And so, but you're right, like, it's a question kind of perspective. I think maybe one way of thinking about it is how many other organisms live in that particular ecological niche. You know, if there's a bunch, even if it's really cold, let's say, it's probably not that extreme. If there's very, very few, then maybe you're more likely to call it an extremophile. That was the beginnings of potentially extremophiles leading to the discovery of life on other planets, as being at least a reason to why there might be life on other planets. I think it's a pretty compelling argument. If we already have them here and they survive without all the things that we say they need, presumably they would be able to survive too. So this is the area that we're in Africa, Ethiopia. And these are the collection sites that the researchers took from. And those, to me, from a genetics perspective, are the coolest extremophiles, because figuring out why they're able to survive in so many different conditions so well, like what is the genetic basis of that, that could be a really, really remarkable thing that maybe we can even like get in on and try to hijack the biology behind it and see if we can adapt it to something else. So again, multiple like extreme conditions and things that would not be favorable for life are happening in these water sources, which is really, really remarkable, I think. And they're able to survive these. So that's just the one gene, the gene structure that usually is rapidly evolving. And that's why one of that's a area where you'll actually actively look at and make sure, is it similar to something known or is it different? And if it's different enough, there's like usual statistical thresholds, then the organism is defined as a different species. Welcome back, Epic with SciAnts. We're going to finish off, you guys, the evening with one more video on just extremophiles. I'm bringing this back. It was good, Chris. I'm pulling us back to extremophiles. We're just going to watch one video on extremophiles before we ran out the evening. Thank you, Sam. That way we're kind of ready to go for next. This is like a teaser for next Saturday. We'll do extremophiles part 3. But if we rabbit hole next weekend as well, that's okay. Because as long as we're learning and having fun and talking science, that's the whole point here, right? Now I have Zen. Nightbot is trying to help Zen. This is again getting back to our idea of it's not just a single like monocell organism that we're having, but rather unicellular, multicellular organisms like insects, fish can also be extremophiles. Welcome back, Smikes. Just in time for the end, good sir. We did make it through a video, Smikes. We did. We actually finished our deep dive, or like our side dive into sharks. Yeah, Smikes, we rabbit hole into hammerhead evolution. Yeah, Smikes. So we're gonna have extremophiles part 3 next week as well. Smikes is like, oh god, Blint, why? It's like, you know, Smikes, we rabbit hole, we do. This probably will be finishing off today's stream, just because Lita should be on her way home soon in a little bit, and then we'll go out and grab some dinner. Thanks for watching SciAnts Media, and be sure to hit those like and subscribe buttons to help our channel grow and support our science outreach and education goals. We can be found on Twitch most days of the week, and be sure to join us here for our next video. Thanks again, and stay curious, everyone.